Systematic sampling, just like other kinds of sampling method, aims to select an ideal range of participants from a target population, and the subjects selected are expected to be able to be generalized to the target population. So how does systematic sampling work? In systematic sampling, the researcher may start selecting samples from a random initial point, but with a fixed periodic interval. The interval is determined by the researcher, which is variously designed according to different experiment conditions to fit the sample size.
Here's an example. Imagine the stickman as your target population. You need to select 10 samples from them with systematic sampling. You set a rule that for every 3 stickmen you will do an investigation. It doesn't matter that you choose to start from the first stickman or the third stickman. What matters is that the interval you set for your sample selecting has to be the same. Therefore, after selecting 10 samples from our target population, we can now look at how our samples' characteristics and quality are. It can be clearly seen that there are only one red stickman, one black stickman, and one blue stickman, but up to 7 purple stickmen were selected. If the samples are used for research, it is possible that the results are going to be biased due to the overwhelming fraction of purple stickmen in our sample. If we see these colors as different characteristics or cultures of human, it is plausible that individual differences and cultural bias would be magnified in this kind of experiment due to the bias of the sample selection. This is a disadvantage of systematic sampling.